Okay, so now we're recording. So good morning, everybody, and welcome to the second talk in this year's series of Talk Grasslands. Today we're going to have two presentations on the topic of flower-insect interactions in mountain ecosystems, presented by two researchers from Milan, Elena Eustacchio and Marco Bonelli. And they're going to give separate presentations, and I think Marco was going to give his presentation first. So now we can listen to Marco's presentation, and then we will change over to Elena. So please, Marco.
Okay, hello everybody. I'm glad to be here, and thank you, Stephen, for the invitation and introduction. I apologize in advance for the poor formatting of some slides, but my presentation file blew up 2 days ago, so I had to start over and finish it in a hurry, but I hope the presentation is still nice. Okay, I try to share my screen. Okay, do you see it?
Yep.
Yeah. Okay, I switch my video because I have bad connection here on mountains in Italy. Okay, do you see it? It's okay?
Yeah, we can see it. If you switch it to the display mode, perfect.
Okay. Okay, so as Stephen said, I will hold the first presentation about our work on flower-insect interactions in mountain ecosystems, while my colleague Elena will hold the second part. And my part, this first part, is focused on the case study of the early flowering in our endemic species, Conversely brevis. Okay, I don't know if you already know this plant, but I introduce it to you briefly. Conversely brevis is an endemic plant that grows in a very restricted area of Southern Alps, above 2,000 meters, about 2,000 meters, 2,200 meters. And this species forms few and also scattered populations that grows often many kilometers apart from each other. Here you can see the map of Southern Alps in Lombardy and Switzerland. And this distribution range was probably shaped by quaternary climatic fluctuations, since, as you can see in this slide, the present population occur only in areas that were ice-free during the last glacial maximum, suggesting a limited dispersal ability. But we also should consider the possible critical role of warm past climatic stages when Alps experience periods warmer than now, with conditions that can be somehow similar to those expected with climate change in the future. Therefore, Conversely can be considered an appropriate model to understand how the past climatic fluctuation shaped the distribution of Alpine plant populations. Regarding the reproduction of the species, instead, it is quite particular because the flowering period occurs very early in the season, immediately after the snow melt, when a few arthropod taxa are active as pollinators, and on the other hand, a few floral resources are available for arthropods. And therefore, it can also be a good model to investigate high mountain flower arthropod interactions, which is our main focus. Indeed, the flower-arthropod interaction, as you know, are essential for the functioning of ecosystems, but the knowledge about them is still limited, especially in mountain environments. I think it can be because, partially at least, because of logistical difficulties in conducting research in these environments, and also perhaps a perceived lack of short-term economic interest in this topic. And also partially because mountain environments are sometimes quite wrongly considered to be unpolluted and undisturbed. So for these reasons, there is little research focusing on these environments. Moreover, we know that mountain environments are expected to be strongly affected by climate change. And this climate change could drive altitudinal and phenological shifts of species, such as pollinator and in general arthropod and plants. But since obviously these shifts may not be homogeneous among the different taxa, some plant-arthropod mismatches could occur, and these mismatches can lead to the extinction, at least local extinction, of both actors, so of plant and arthropods. And this local extinction on this strong reduction can have cascading effects on the ecosystems. In particular, it is known that the early season, when Androsace brevis flowers, is a very critical moment for possible mismatches. Therefore, in this context, we have undertaken a project that aimed to assess the genetic structure of Androsace, to unravel its reproductive strategy, and especially to investigate the flower-arthropod interactions in which it is involved as a model for early season interactions in high mountain environments. And obviously, all these points are quite interrelated, since the genetic structure of Androsace is shaped also by arthropod pollination, obviously. So all these points are not separated, but are quite interrelated. Okay, to assess the first two, to achieve the first aim, we sampled 8 populations across the species range, and overall we genotyped 350 plants with nuclear simple sequence repeats that are molecular markers suitable to assess the genetic variability both within and among populations. Okay, as you can see in this slide, by the percentage of polymorphic loci, we observed a high genetic diversity within populations, but lower in northern ones, with a marked lack of variability in the smallest population, that is Valmasino, as you can see here. Moreover, we assessed a low number of clones in most of populations, and these clones usually were limited to neighboring plants. This indicates a marginal role of vegetative reproduction, because with the main role of vegetative reproduction, we would expect to find many clones. Instead, the low number of clones is an indication about a low level of vegetative reproduction. Finally, these results, together with a low inbreeding coefficient, indicate or just suggest that Androsace is mainly an outcrossing species. This is about the diversity within populations. Regarding instead the genetic differentiation among populations, as you can see by the PCOA, there is a clear separation of northernmost populations from the southern ones. And this separation was also highlighted with another method, with the Bayesian model-based clustering method implemented in structure. So we have these 2 clusters of populations. As a next step, now we are trying to infer the past population history with do-it-yourself ABC, a software, to understand which complex evolutionary scenarios have led to this present situation. However, we already estimated a gene flow among populations, and considering the 2 identified clusters, the north and the south one that I show you, there seems to be a higher gene flow from north to the south than the contrary, as you can see it here. Indeed, in the investigation area, the most frequent and strong winds blow from the north to the south. So it may be possible that this gene flow is due to pollen carried by arthropods that sometimes can be carried by the wind from north to the south with higher currents. However, No information was available about the pollination biology of Androsace, and also no information was available about its possible pollinators, and in general about its interactions with flower visitors. So we decided to start our research investigating these aspects. As a first step, we conducted an experimental exclusion of potential pollinators to investigate if they are necessary for the fertilization of these species. And we observed no difference in flower number among control and excluded and treated plants, while, as you can see here in the plot, we have a significant difference in seed-flower ratio with 0 seeds for excluded plants. So this is a strong indication that Androsace brevis needs pollinators for its fertilization. Indeed, we also observed that the color of flower mouth in Androsace are linked to the repairing stage of anthers. And this is not only a feature of Androsace brevis, but is common through the Androsace genus. And this may be a signal about the quality and quantity of floral rewards for potential pollinators. So is another indication about the role of pollinators. However, continuing to exploring the flower morphology, regarding the potential pollinators, this morphology does not provide any indication about possible pollinators, because it does not show features that can select specific insects. Because the presence of anther and stigma, as you can see here, less than 1 millimeter below the corolla mouth, so below the entrance of the corolla, and also a diameter of the mouth quite wide, can allow many flower visiting insects to reach nectar and pollen and also to pollinate. Moreover, this quite relatively wide mouth also could allow many different insects to use the corolla tube for sheltering, for instance, or for other activities. Because, as you know, the insects does not use flowers only for trophic activities, but for many activities like sheltering, like hunting, like mating. So this flower morphology didn't give us strong indications about its relation with pollinators and in general flower visitors. Therefore, we decided to investigate the diversity, the role, and the behavior of possible Androsace flower visitors by integrating 2 methods. We integrated in particular the traditional manual sampling, so capturing the insects that visit the cushion, the flowers. And this method allows to obtain a fine-scale identification of sampled arthropods, obviously, and also to evaluate which taxa actually carry the pollen of the plant. With observation, instead, are a less invasive method, and they can allow to spot elusive taxa, but especially the strong feature of this method is that it allows to assess the arthropod behavior. So we integrated these methods to investigate the arthropods visiting Androsace, but it can apply to all high mountain flowering plants. Okay, we performed the first method, manual sampling, in 2 study sites, which are in different areas, but at the same about the same elevation. And flower visiting arthropods were sampled for 5 years during the brevis flowering period. As I told you before, this flowering period is very short, about 2 weeks between the end of May and the beginning of June. So in this period, we sampled the arthropods visiting the flower according to the time and the observation plot method, and we collected all the arthropods visiting the flowers in ethanol. Then these arthropods were identified both by morphological traits, by expert taxonomists, and by barcode DNA sequencing. And we arrived in most of cases at species level and always at least at a family level. Instead, at order level, here I present you the order level because we cannot enter in detail to species level. We will see something on family after. But at order level, as you can see, insects belonging to the order Diptera and Hymenoptera were the most abundant. With Diptera representing almost half of the collected specimens and Hymenoptera more than 1/3. So now we see the family a bit more in detail. Regarding Diptera, most of them were flies belonging to the family Anthomidae. Anthomidae is a family of Brachycherus Diptera, and they visit flowers possibly because of trophic activity, as you can see in this picture. While other family was less than 15% each, and were mostly other Diptera Brachychera. Regarding Hymenoptera, instead, the most represented family was Formicidae, the ants. In particular, we found Quidachus and Nectarivorous species that are known to forage on ground and plants, so nothing strange under this point of view. But moreover, we detected many parasitoid species, mostly braconids, which adults can feed on nectar as adults, however, for these parasitoids, both Braconidae and other family, a curious fact is that we sampled only females. So it is also possible that the presence of these parasitoids was linked to search for some host to lay eggs on the flowers, perhaps. Instead, visits by Apoidea anthophila, that are the bees, were very rare. And this is quite peculiar compared to the later season communities that are also main studies. So these three are the difference that we found in early season community compared to studies about later seasons community, and this is a quite peculiar community. We published the paper about this if you're more interested in detail of this community. Okay, so we assessed the Androsace brevis flower visiting community, but obviously not all the visitors are pollinators, because as you know, not all flower visitors can actually carry the pollen. And despite this, many studies about pollination and pollination networks are currently based only on the observation of flower visits, while few have tested whatever the flower visitors actually carry the pollen of the plant. So to discriminate the actual pollinators of Androsace, we performed quali-quantitative palynological analysis of pollen grains carried by sampled arthropods. So what does it mean? That we collected all the pollen grains from the bodies of the sampled arthropods, and then we collected the pollen from the flowered species in the investigated area during our study period, so Androsace, but also other plants, as you can see here, few plants, because it's I remind you that is early season. And then we compared all pollen grains found on the arthropods with the pollen grain of the library that we realized with the plant flowered in the area. And we counted all the pollen granules, and on which arthropod taxa we found Androsace pollen. As you can see here, on the material collected the first year, the largest share of Androsace brevis was found in Hymenoptera apoidea, in particular bumblebees, with a minor share on Diptera anthomidae, these flies. The second year, Androsace brevis pollen was found only on Diptera anthomidae, and the third year only on Hymenoptera apoidea, in this case Alictidae, another family of wild bees. So It seems that pollinators can vary annually, probably due to the climatic or environmental condition of the year, but we can conclude that Diptera, Antomide, and Hymenoptera apoidea, so wild bees, we did not record Apis mellifera, so all these were wild bees, are the main pollinators of the Androsace brevis. And it is interesting to notice that bees, despite being very uncommon flower visitors in our context, as I told you before, can still be relevant in pollination due to their very efficient pollen carrying structure. So even if the number is low, the role in pollination is very important of wild bees. Moreover, we counted all the pollen granules carried by arthropods, as I told you before, and so we were able not only to identify the pollinators of Androsace, but also to obtain pollination networks based on the actual pollen carried by arthropods, not only on flower visits, for all the context. So here you can see the network from arthropods' point of view above, and this is the line above, the network from arthropods' point of view. And this means that the line thickness, this yellow line thickness, represent the percentage of pollen of different plant taxa found on an arthropod taxon. While below, you can see the same network from the plant's point of view that indicates on which arthropod taxa we found the pollen of a specific plant taxon. I do not enter now in the detail because it will be quite long, but with this innovative approach, we were able to identify the floral preference of the arthropods. And we also assessed that Hymenoptera apoidea and Diptera Antomide are main pollinators, not only for Androsace brevis, but also for other Entomophilus species, as you can see from the plot above with the plant's point of view. Finally, despite the moment, so despite the early season, the plant pollination networks seems to be quite complex and well structured, but the network analysis is still in progress, so I will not enter in detail. Then we also tested the effect of environmental variables on flower visitors, in detail, in particular on flower visitor presence and abundance. As you can see, the temperature has a significant positive effect on both presence and abundance, at least at the recording conditions, even if we cannot rule out that higher temperature, as one expected in future with climate change, could negatively affect the flower visitors' activity, since it is also possible a non-linear response of arthropods. But in our working condition with a quite low temperature, the effect was positive. While regarding the daily pattern of flower visitors' activity, we observed a significant effect of the quadratic time. And so this, what does it mean? This means that we observed a bimodal pattern with morning and afternoon peaks of presence and abundance. And this pattern has been already observed in some other studies for Berticeran flies and also for some bees, ants, and if I well remember, also for some beetles. So it's quite a confirmation. Also, if this pattern can be influenced by climate condition, therefore also this aspect could be potentially impacted in future by climate change. On the contrary, as you can see here, we did not observe a significant effect of the wind speed, and also we did not observe a significant effect of the number of flowers per plants. All this data I showed you was about the whole community, so considering all the flower visitors. Perhaps in future with more data, it will be interesting to test the effects of these variables also on the single taxon, because obviously different taxon can react differently to environmental variables. But with these 4 years and with not a high number of visitors, because the number of visitors is limited because of the moment of the early season, we are not able to perform for now analysis on single taxa. So this is a general framework. In future, it will be interesting to deepen this aspect. So manual sampling allowed us to describe the flower visiting community, to identify the main pollinators of San Rosace, to have new insights on the early season plant-pollinator network, and also to understand how arthropods activity is impacted by environmental and temporal variables. But obviously, this method cannot give us information about the behavior of flower visitors, because with manual sampling, as soon as an arthropod arrived on the flower, we captured it. So it is not possible to evaluate what they do on the plant, on the flowers. So as I told you before, we evaluated the activity of flower visitors with video recording sessions. We performed a total of more than 100 hours of footage. The third year is still current under analysis. Then we watched and analyzed them through behavioral observation software. Do you still hear me? Because
yes, we can hear you and see you.
Okay, because I have bad connection, it gets stuck for a while. Okay.
No problem.
So I was saying, we used behavioral observation software, BOIS, and we developed an histogram, a de novo histogram, to investigate the activity and time budget among taxa and to understand how and why they interact with the flowers. Almost all the flower visitors were identified at order or lower taxonomic level, and this is not easy by a video, I show you. And then we evaluated their ecology. Here I will present you just some example to show you the potential of this method and not all the whole analysis, because it will be very long. In this plot, you can see the taxa we identified as flower visitors and their entrance frequency. The entrance frequency is the percentage of observed subjects entering the corolla tube. And in particular, okay, sorry, sometimes it gets stuck. And in particular, we performed a statistical analysis on the 5 taxa with at least 15 entering subjects in the videos. And these taxa were Coleoptera, so the beetles, Diptera brachycera, so suborder of flies, Hymenoptera poidea anthophila, the bees, and other Hymenoptera that are wasps, and in this case were mostly parasitoids. And finally, Tisanoptera that are the thrips. Okay, here you can see plots of post-hoc pairwise comparison for different behaviors. Each red segment in these plots represent the confidence interval for difference in means between the 2 compared taxa. To make it simple, because it is not very simple if you don't have familiarity to this plot, but to make it simple, if the red segment crosses the vertical line, there is no significant difference. If the segment is on the right, the first taxon wins the comparison. If it is on the left, the second taxon wins the comparison. It could be quite tricky. So to increase the readability of the plot, I just put a clip art representing the winning taxon on a significant difference, so it's easier to understand. As you can see, so we can immediately discriminate 2 different patterns in the behavior of flower visitors. because Hymenoptera poideantophila, so the bees, and Diptera brachycera visits many flowers, but spend little time on them. And this, together with very high entrance tendency, especially for bees, confirm their main role as pollinators, as we assessed with manual sampling. And on the other hand, beetles, thrips, and parasitoids, wasps spend more time on flowers, but visits less flowers. Thus, they can be less likely efficient pollinators. However, within this second blue group, we can discriminate again two patterns. Beetles and thrips, as you can see here, spend longer time inside the flower, feeding on the flowers, and in case of thrips, also sheltering and mating in the corolla tubes. Instead, the parasitoids spend less time inside the flowers and have a longer latency. What is the latency? The latency is, we mean with latency, the time before entering the flowers. So parasitoids spend a lot of time on flowers before entering the corolla tube, where they can then feed on nectar. I remember you the fact that for parasitoids, we recorded only females by manual sampling. So this long latency, this long time after this long time out of the corolla tube can be linked to host research on flowers, to search host to parasitize. So the video observation confirmed and supported the main findings obtained with manual sampling, but also gave us further insights on the behavior of flower visiting subjects. And this is a poorly investigated topic. So we provided information that we think it can be quite innovative. So after this long presentation, I just leave you some take-home messages. We assessed that there is a genetic separation between southern and northern population of Androsace, with the latter showing less genetic diversity. And the species is mainly outcrossing and need pollinators for reproduction. And Diptera antomide and Hymenoptera poideantophila are important pollinators of this species and of other high mountain early flowering species. Temperature and daytime can impact on the presence and abundance of flower visitors. Despite the peculiarity of the considered period that I remember used the yield season, the pollination networks are quite complex and well structured, and Androsace is visited by different taxa with different ecological roles and behavior, and these taxa exploit the flowers in different ways. Moreover, we developed with Androsace a set of tools that can be useful to investigate flower-arthropod interactions, also in the broader context of high mountain ecosystems, not only on Androsace, as Elena will show you now. Thank you for your attention.
That's great. Thank you very much, Marco. Let's go straight over to Elena's presentation, and then we'll take all the questions at the finish.
Yeah. I try to interrupt my sharing.
It should offer you to stop sharing, and it will take it anyway if Elena switches hers on.
Okay, I think.
Okay.
I did it.
Okay. Hello.
Hi, Elena.
Okay. Okay. Okay, can you hear me?
Yes.
And
Clear.
can you see the presentation?
Everything's fine.
Okay. Okay. Hi, I'm Elena, and I'm a PhD student of the University of Milan in Italy. And my talk is focusing again the flower insect interaction in mountain ecosystem. And just a brief introduction, because Marco said all the things before. As we know, plant and arthropod interact with each other, creating complex networks such as those related to pollination. So these interactions are exposed to natural and anthropogenetic traits, as we know, mainly related to climate warming that is associated with phenological and altitudinal adaptation in a wide variety of organisms. And as we know, plants and arthropods could respond to climate warming in different ways, and so that mismatches could occur, resulting potentially in lower fitness for both parts. But in addition to the climate warming, in the last decades, arthropods are decreasing in biomass, and the local extinction rate is increasing for many species. Also, for the other decline factors that we can see in alpine environments. One of these are the type of agroecosystem management that could affect the arthropod communities directly using the insecticides, or indirectly using by the alteration of plant communities caused by the change of land use using herbicides or fertilizers. Moreover, as Marco said before, there is a poor knowledge of floral visitors communities of agroecosystems in alpine environments. Our aims in this case are quite similar to those explained by Marco, are to provide a description of plant arthropod networks, this time along an altitudinal gradient and in different type of land management. Moreover, to identify the focal plants and animal species responsible for the network stability and organization, and to study the behavior of flower visiting arthropods on flowers. We studied on the southern parts of the European Alps. Alps are a biodiversity hotspot, especially grassland ecosystems. And on the Alps, the knowledge on flora and fauna is high. In particular, we are studying in Martello Valley, that is a valley located within the Stelvio National Park, that is a park located in the eastern part of the Alps. We selected 14 plots, 6 in Low Valley, 4 in Mid Valley, and 4 in High Valley. In particular, we worked we selected 2 pastures and 2 high meadows all along the altitudinal gradient. Of course, the high meadows were replaced by high altitude grassland in the higher part of the valley. And in addition, we selected also 2 apple orchards, because this crop is a very common environment within the Stelvio National Park. Our sampling design was focused on 3 plant families, not just on one plant as you saw before. So we selected Asteraceae, Fabaceae, and Ranunculaceae. First, because they present a different flower morphology, as they are more attractive for different type of arthropods. And they are present all along the altitudinal gradient, and they flowers in all the selected plot and for all the season that we carried the sampling. The sampling has been done from 9 a.m. to 6 p.m., excluding obviously rainy days, from May to August 2021 and 2022. Our methods were the same that we used for Androsaceae brevis. We used manual sampling of arthropod visiting observed flowers through timed observation method. As Marco said before, it is a traditional method that allows a fine-scale taxonomic identification of the sampled arthropods and allows to evaluate which taxa carry polygrains. At the same time, we conducted a simultaneous video recording session on the same plant species we were observing in that moment, it is an innovative, non-invasive method that allows to describe behavior and activity of a flower visiting arthropods on plant. We studied all the flower visiting arthropods that touch the flowers, but as we know, the main studied pollinator taxa are Hymenoptera, and in our study we kept wild bees and honeybees separate, Dipteri, Syrphidae, and the Lepidopteri loparocera. At the end of the 2 years of field work, we can see that we sampled 399 specimens, and more than half of the specimens were Syrphidae, followed by Apoidea and Lepidoptera. Syrphidae are very common and abundant both in 2021 and 2022, especially in June and July, while as well as wild bees. Also, if in June 2022 they were very little represented, while Apis mellifera and Lepidoptera present a different trend. In 2021, sorry, Apis mellifera was very abundant at the beginning of the season, and then its abundance decrease, while in 2022 it present a peak in May and August, perhaps for the different meteorological condition. Also, Lepidoptera in 2021 were very more abundant in the late season, while in 2022 were little represented, but all along the season. If we observe their distribution along the altitudinal gradient and between different land management, we can see that Syrphidae are very represented, very abundant, mainly in mid and high valley, while Apoidea are very abundant just in the low part of the valley. Especially wild bees are very abundant in the pastures, perhaps for the limited presence of heavy vehicles in this type of environment, because as we know, most of wild bees nest underground, and heavy vehicles could stress them in this delicate period of their life. As Marco said before, we conduct the palynological analysis. We separate pollen load from the specimen, and we for now analyzed only the pollen load carried by bees. So pollen was removed from bees' bodies, and the pollen material from bees belonging to the same family separating honeybees, agroecosystem and altitude was grouped and dried from conservation ethanol. We analyzed the pollen grains carried by these samples in 2 ways: by morphological identification and molecular identification, using light microscope and DNA metabarcoding. By this way, we identify the networks for all the ecosystems and along the altitudinal gradient. Here we can see the relative abundance of plant families present in the pollen load of Apoidea families, and with the columns with the two colors, you can see the pollen grains belonging to the families on which we carried the sampling, so Asteraceae, Fabaceae, and Ranunculaceae. Especially Fabaceae and Asteraceae are very abundant in the pollen load, but they are not the only families that we observed. You can see that especially in low valley, there is a lot of pollen of, for example, Boraginaceae and Dipsacaceae, while in High Valley there is a lot of pollen belonging to Campanulaceae and Ericaceae, as well as Plantaginacea and so on. But the most studied pollinator taxa, such as Aquidea, Syrphidi, and the Lepidoptera, represent just the 9% of the flower-visiting arthropod that we collected on the flowers. In fact, as we can see here, about the 0.5% of the sampled arthropods belongs to the Tisanoptera order, followed by Diptera, Hymenoptera, Coleoptera, Hemitteri, and then Lepidoptera, Aranei, Collemboli, Acari, Orthopteri, Dermateri, and Neuropteri. As we can see along the altitude, the elevation gradient, Tisanoptera are very abundant, especially in low valley, while in mid-valley, Diptera become very abundant, while in high valley, Coleoptera, Diptera, and Tisanoptera are quite similar between each other. If we observe their distribution among land management, Tisanoptera are very abundant in all the land, in all the environment we studied in low valley, and they return to be very abundant also in pasture in high valley, while in mid valley, Diptera are very abundant both in pasture and high meadows, while in high altitude grassland ecosystems become very abundant both Coleoptera and Diptera. We can try to give an explanation of these different abundances in the different environment. Regarding Tisanoptera, we know that this is a polyphagous taxon known for the damages that it could make to crops, and some genera such as Frankliniella and Thrips are known to be important pollinators for these ecosystems. Our samples will be identified soon in order to discover if these two genera are present in our studied communities. Moreover, Tisanoptera have a peak of activity in meadows below 1,500 meters, usually in summer, when the floristic diversity is high, and Tisanoptera shows a low mortality. In this environment, they find an optimal condition of temperature, light, and humidity. Moreover, we know that some species are important for the stability of mountain meadows ecosystems, occupying an important ecological niche in the structural net of the whole environment. We found also Tisanoptera also above 1,500 meters, and they seem to be related to grazed areas. So pollen analyses are essential to discover if they are also pollinators. Then we discovered a lot of Diptera, and as reported by literature, Diptera increased their importance as flower visitors with the altitude. And in 2021, we collected mainly Antomidi, Chloropidi, Muschidi, and Syrphidi. And we can see that especially Muschidi increased their abundance with the altitude, while Antomidi, Chloropidi, and Syrphidi tend to decrease their importance, and this is what we observe both in literature and in field. Regarding Coleoptera, as reported by literature, they decrease their importance as flower visitor with altitude. And we know that they are one of the main pollinator order below 1,500 meters. But we discover a lot of samples belonging to the Meliridae family. This family is represent an important pollinator family in the plains for example, Leucoium estivum. In our case, they are feeding and mating on the Leucanthemopsis alpina at 2,700 meters in the high altitude grassland ecosystem. And they are pollinators. We have to discover it doing palynological analysis. Okay. Moreover, we analyze the video recording, the video that we made on field with a software, Boris, that allows to create an ethogram to study selected behavior that we want to analyze. In particular, we studied the frequency of contact with reproductive part of flowers done by main pollinator taxa. In particular, we studied Apoidea, Syrphidi, Brachycera, all those Brachycera that are not Syrphidi, Lepidoptera and Coleoptera. As we can see, Apoidea, Syrphidi and Lepidoptera made a lot of contact with reproductive parts of flowers rather than the uncontact, okay? While Brachycera and Coleoptera made less certain contact with flower reproductive parts and a lot of uncontact. So they visit the flower but don't go for their search of nectar or pollen. This trend we can see this trend also for the time spent on flowers by these main pollinators taxa. As we can see, Coleoptera and Brachycera stay a lot of time on flowers. As we can see in the video, Coleoptera could stay on flowers not for searching nectar and pollen, but for feed on petals or for I don't know what this one is doing on the flowers, but surely is not interested in pollen. And while Diptera, Syrphidi, Apoidea, and the Lepidoptera stay less time on flowers because they search just flowers with pollen and nectar, feed on them, and then go away. So in conclusion, orders abundance changes as function of season, altitude, and different type of land management. Tisanoptera with the increasing altitude seems to be more related to anthropogenetic habitats, where could play an important role in the network organization. Diptera and Coleoptera increase their abundance with altitude and spend more time on flowers than Apoidea, Syrphidi, and Lepidoptera. And in conclusion, the role of pollinators is potentially played by more taxa than those commonly studied and protected. And thank you for your attention.
Great, thank you both very much. Does anybody have any questions for either Marco or Elena? I'd like to ask Marco anyway, something about the phenology.
Yeah.
Do you think with that species of plant that's the trigger for flowering or for the timing of the flowering is the snow cover? And if so, does that vary very much in that region? Because here in Finland, for example, we see that the snow cover in the south of the country is completely erratic, but in the north of the country, it's not changed very much. I know there's altitudinal effects there. So, like what I'm thinking is presumably the snow cover, the disappearance of the snow is what gives the signal for the flower to start producing flowers.
But
and probably also the pollinators as well.
Yeah, but it is known that somehow plants can anticipate more than pollinators, because when there is no cover, androsyche can flower, while some pollinators need a period in days of overwintering. So they did, for instance, a study in Japan where with a simulated anticipation of snow melt, the plant was able to anticipate, but the pollinators were not able to anticipate so much. So it is possible that the plant flowers if the snow melt anticipate, but there are no pollinators. So this is the very critical point of early season for mismatches is indeed related to snow melt.
Yep. And I was also wondering about the plant species on the basis of the genetic studies you've done. Do you think that the population is becoming fragmented and isolated, or is there not so much change in that respect?
Yeah, we observed by our estimation from north to south 9 events of gene flow for a generation, but this plant lives quite long. It can live. We are not sure data about this because they are not in literature and it is not easy to estimate, but we think it can live like 30 years perhaps, or 15 years. So the 9 events in a generation can seem a lot, but are not 9 in 1 year, but can be 9 events of gene flow in 30 years, for instance. So yes, it is quite separated. But the interesting fact is that the north and south populations are more separated than east and west. But we have a lake in the middle. So it's somehow surprising because we thought to have a separation because of the lake too, but we did not observe this separation. So probably it can be related also to historical facts and to the last glacial maximum.
Yeah. Okay, does anybody else have any questions? I'd like to ask Elena something as well. Regarding the video studies, you were using the length of time that an insect was in the plant as an indicator of whether it was pollinating or not. I'm wondering whether it's presumably not going to be perfect that also parasitoids that are seeking prey, they will be affected by different factors to pollinators. So I guess that would also overestimate some types, so that they would spend a long time on the flower, but still maybe not pollinating. I guess what I'm wondering is if this is like a nice first study about this, but do you think there is things that you would change in the methodology to make it maybe even more accurate?
Okay. Yes, we with the videos don't we just suppose if visitors are pollinators or not, because of course with video you can see if they are carrying pollen or not. So for this we conduct both videos and sampling, because with video we just wanted to see if they contact the anthers. So if they touch them, we suppose that they carry pollen, but yes, they are just hypotheses. We construct the network just only on the sampling, on the sample arthropods. Yes.
The other thing I was thinking, that there's also a lot of species of Muscidae, for example, and also Scaraboidae, which have very, very hairy bodies, and pollen does attach quite well to it. And though they're not generally considered as pollinators,
Yes, in fact, most of studies are based on Hymenoptera and Sylphide and so on. But regarding Coleoptera, we see that a lot of them carry pollen grains because they are they spend a lot of time on big flowers, especially Asteraceae or yes, also Ranunculaceae, because they have a big flower and they can fly on them without jump down.
Yep. Yes.
Yes, they are very funny to see.
Yes. Another thing I wondered about the plant species that Marco was working with, are there other pollen sources at that time of year early in the season, or is it fairly exclusive?
No, there are about 13, if I well remember, flower species, and I think 13, if this information is supplementary of the article, I'm not sure, but there are a few other plants flowered in a radius of 1,100 meters. And for this reason, that the number of plants is limited, we were able to do a pollination network, recognizing all the species, because later in the season it will be very difficult to. But here we were able to identify all the flower species because we did a check of.
Good. Okay, well, it looks like there are no other questions. So I'm going to stop the recording now. So